Chapter 11 discusses innate and adaptive immunity. So the definition of an immune response is the collective coordinated response of the cells and molecules of the immune system. And within that immune response, we have two separate types of immune defenses that work together to provide the entire immune response. So we have our innate or our non-specific immunity. This is the natural resistance which people are born with that responds to infectious disease. And then we also have our adaptive or our specific immunity, which is our second line defense. It responds less rapidly than the innate immunity, but much more effectively. So this table is from your book and would be a good spot to review when considering the differences between these two types of immunity, both innate and adaptive. So just a little spot that you can go to kind of really see the differences in one really nice table. Okay, so the cells of the immune system. We have lymphocytes. Lymphocytes are cells that specifically recognize and respond to foreign antigens. We have B lymphocytes and T lymphocytes, and we'll talk about each of those. And then we also have accessory cells. These include things like macrophages and dendritic cells. They function within the immune system as what we call antigen presenting cells or APCs, and they process complex antigens into something called epitopes. And this processing and presentation is required for the activation of lymphocytes so that they can respond to those foreign antigens. Macrophages as well are responsible for clearing bacteria while they're presenting those antigens for lymphocytes to kind of react to. Macrophages also secrete substances that initiate and coordinate the inflammatory response and that activate lymphocytes, so things like different chemical mediators. The dendritic cells are really kind of a bridge between the innate and adaptive immune systems. They function as some key antigen presenting cells or APCs that initiate that adaptive immunity, and they're responsible for processing and presentation of antigens to the lymphocytes. So different mediators of the immune system. There are the cytokines, which are those soluble proteins secreted by cells of both the innate and adaptive immune system. So cytokines are short-acting, biologically active soluble substances. They're an essential component of host defense mechanisms, and they are the primary means by which cells of the innate and adaptive immune systems communicate. There is a table in the reading that discusses all of the functions of the different cytokines in both innate and adaptive immunity. I don't expect you to memorize all of these, but do take a look so that you can really understand the wide range of the things that those cytokines control. The table is table 11.2. So cytokines are interleukins or ILs, interferons or IFNs, and tumor necrosis factor alpha, so TNF alpha. There are also, of course, the chemokines, and those are a specialized type of cytokine that stimulate the migration and activation of immune and inflammatory cells. And finally, the colony stimulating factors. These are also a type of cytokine that stimulate the growth and differentiation of bone marrow progenitors of immune systems. So colony stimulating factors encourage hematopoiesis, which produces large numbers of things like platelets and erythrocytes and lymphocytes, neutrophils, monocytes, and eosinophils, basophils, and natural killer cells. So all of those different kind of cells that are necessary to mount a really good immune response. Colony stimulating factors are largely a type of cytokine that are taking care of that stimulation. So a few more things about cytokines, just some general properties. So again, they're released by the cells of both the innate and adaptive immune system to regulate the action of these cells. They bind to receptors on target cells and activate those intracellular processes. When they're released in a really brief, self-limited manner. Remember, we said that they're really short-lived kind of chemicals, and we rarely store any type of cytokine. And that is by design so that their action can really only be very brief in nature. We don't want kind of this prolonged and unchecked kind of activation of cytokines in general. It's really important to have that self-limited nature. And malfunction of that particular defense system that allows cytokines to kind of continue their action can lead to different diseases that are inflammatory in nature. Things like psoriasis or asthma are linked to malfunction of that system. But cytokines can initiate this cascade of reactions where one cytokine influences the synthesis of another and then the action of another. So while one cytokine is kind of self-limited and dying off, while it was active, it stimulated a cascade of another. And that's another kind of self defense sort of mechanism to, again, make sure that those cytokines don't just continue to act without control. So we're going to start with talking about the first type of immunity, which was that innate immunity, that immunity that everybody's kind of born with, that's very nonspecific, that starts acting really quickly in reaction to kind of a foreign substance. So the different components of innate immunity, epithelial barriers. So epithelial barriers are those physical, mechanical, and biochemical barriers that are found in all portals of entry. So places like the skin, the respiratory tract, the GI tract, urogenital tracts. So the epithelial cells found at those portals of entry can secrete things like enzymes and proteins that destroy invading microbes. They also contain things like simple chemicals like salts or acids that can inhibit the colonization of microbes at those epithelial portals of entry. Some cells of these linings can do things like secreting mucus that can trap pathogens. Some epithelial cells have things like cilia to try to trap microbes. So all kinds of variation and action of epithelial barriers to help prevent invasion of microbes. There are also, of course, those phagocytic cells, so things like neutrophils and macrophages, those neutrophils being the most abundant granulocyte in the body. They're phagocytic, meaning they can engulf microbes. They can migrate throughout the body by this like amoeboid type of movement. And they are really our very early responders to foreign invasion in the body through that innate immune system. Macrophages are released by the bone marrow and they phagocytize foreign substances and then go ahead and collect any cellular debris at the site of inflammation. They have much longer lifespans. They are really helpful for limiting the spread of infection until those adaptive immune responses can become fully activated. They also play a role in presenting antigens for the initiation of the adaptive immune response. Dendritic cells, or DCs, are leukocytes that act as kind of the bridge between innate and adaptive immune systems because they are also largely responsible for processing and presenting antigens. NK cells, or what we call natural killer cells, have the innate ability to spontaneously kill target organisms. So they also work to help limit the spread of infection and assist in the development of adaptive immune responses because the NK cells can produce the chemical mediators of cytokines. Let's see, opsonins coat particles and enhance their recognition and binding by phagocytic cells to microorganisms. There are also acute phase proteins like CRP, C-reactive protein, that get released in response to injury and inflammation. They help to enhance the binding of phagocytic cells to invading microorganisms, and they also activate something called the complement pathway in immune function. And the other big thing that many of these cells do and what happens in innate immunity is one of those things we've already talked about, which is that induction of the inflammatory response. So the innate recognition system. So the innate systems can differentiate self from non-self, and that's an important piece of the immune system, right? We want to make sure that our immune system is functioning to attack foreign substances and not attack our own cells. And we talked a little bit about what happens when that doesn't go right, but we should be able to differentiate self from non-self. And the PRRs, or pattern recognition receptors, are these sensors found on the cells of the innate immune system that are largely responsible for this innate recognition system function. They are what we call host sensors, so sensors on the host cells. Invading pathogens contain structures in their cell membranes called PAMPs, or pathogen-associated molecular patterns. And those PAMPs are recognized by the cells of the innate immune system. When PRRs, or pattern recognition receptors, come in contact with PAMPs, they trigger this pro-inflammatory and antimicrobial response that includes the release of things like cytokines, chemokines, and cell adhesion molecules that start that whole inflammatory and innate immune response. There are also TLRs, or toll-like receptors. TLRs bind to the PAMPs and also play a role in things like wound healing, tissue repair, and tissue regeneration. So a little bit about the complement system within the immune response. So the complement system is composed of groups of circulating proteins that are inactivated precursors for this immune response. When they're activated by an immune response, they function as, their functions are opsonization of invading pathogens. Remember from that earlier discussion that that's this kind of coating of pathogens that make them easier for things like phagocytic cells to kind of recognize and engulf. They also help with the migration of leukocytes to an area of injury. They help with the initiation of the local inflammatory reaction and also the lysis, or destroying of pathogens. The complement system will function within both the innate and adaptive immune responses. And another one of its primary functions is the promotion of inflammation and the overall kind of destruction of microbes. So the adaptive immune system is the final line of defense against infection, and it's activated once the innate immune response initiates that inflammatory process. So the adaptive immunity is able to recognize and react to many microbes and non-microbial substances. It has the ability to distinguish among different, even closely related microbes and molecules, and then it can remember those pathogens and quickly produce like a heightened immune response if that pathogen is encountered again. Lymphocytes and their products are the primary cells of the adaptive immune system, and the way that they primarily work is through the identification of antigens. So there are two different types of adaptive immune responses: humoral immunity and cell-mediated immunity, or cellular immunity. Humoral immunity is mediated by molecules in the blood, so these are traveling through the bloodstream kind of at all times, and these are our B lymphocytes. So humoral immunity, humoral equals blood. B lymphocytes are found in the bloodstream. Humoral immunity is our principal defense against extracellular microbes and toxins. And then the cell-mediated immunity is mediated by our T lymphocytes. And T lymphocytes defend against things like intracellular microbes, so things inside the cell as opposed to extracellular like humoral immunity. So largely things like viruses or anything that has taken up residence inside of a cell is what the T lymphocytes will destroy. So a little bit about antigens and antibodies. So antigens are substances foreign to the host that can stimulate an immune response. And antibodies are things that recognize antigens. B cells, or B lymphocytes, which are part of that humoral immune system, secrete antibodies. We call these antibodies Igs. In response to antigens, leading to the destruction of that invader. Antigens are recognized by specific receptors that are present on the surface of lymphocytes and by the antibodies or Igs secreted in response to the presence of that antigen. So antigens possess immunologically active sites called antigenic determinants or epitopes. These are smaller discrete components of the antigen that have these unique molecular shapes, and they can be recognized by and bound to specific Igs or antibody receptors found on the surface of the lymphocytes or by an antigen binding site of a secreted antibody. So you can see here in the diagram, here's my antigen, that foreign substance. And on the surface of those antigens, we see these antigen determinant sites or our epitopes kind of displayed on the surface. So you can see those kind of here. They almost look kind of like a little, lock, right? And then those things are specifically bound to by these different antibodies, right? Antibody A, B, C, or Igs that are secreted by the B lymphocytes. So antibodies secreted by B lymphocytes attach themselves specifically to these epitopes that they recognize on the surface of the antigen or foreign substance that has invaded the host. So these are just some types of antigens, and these should look very familiar as our different types of pathogens, things like bacteria, fungi, viruses, protozoa, parasites, and different non-microbial agents or toxins. So again, here are the cells of the adaptive immunity. We talked about lymphocytes a little bit already. We'll say a few more things about them. So those B and T lymphocytes. The APCs are the antigen producing cells. And then we also have some effector cells. So here are our lymphocytes, a little bit more about the different types. So 36% of the total WBC count are lymphocytes, and they are the primary cells of the adaptive immune response. They arise from the lymphoid stem cells in the bone marrow. They are the only cells capable of recognizing organism-specific antigens on the surface of pathogens, giving that adaptive immune response its memory. Lymphocytes are distinguished by their function and response to antigens, as well as their cell membrane molecules and receptors, the types of secreted proteins, and their tissue location. So once the invading substances or organism has been removed from the body, the lymphocytes can remember the presenting antigen and can respond rapidly during the next encounter. And so these lymphocytes are called memory T and B lymphocytes. So again, B lymphocytes are the humoral immunity. They have the ability to remember, they have memory. These B cells mature primarily in the bone marrow, and they secrete antibodies. T cells are part of that cell-mediated immunity. They also have memory. They mature in the thymus rather than the bone marrow. And in the case of T cells, they don't secrete antibodies. Instead, the entire cell is involved in the immune response. So you can see here in the bone marrow, we have those pluripotent stem cells that will produce lymphocyte stem cells. And some of them will travel to the thymus where they mature into T cells. And some will travel will stay in the bone marrow and mature into B cells. Okay. Then both types of cells, T cells and B cells, travel to the lymphoid tissue. So these are places like the lymph nodes, the spleen, mucosal tissues, blood, and lymph systems, so that they can either be stored or travel throughout the body for use if there is an encounter with an invading antigen. So functionally, there are two types of immune cells, regulatory cells and effector cells. The regulatory cells assist in orchestrating and controlling the immune response. The regulatory cells assist in pre-planning and controlling the immune response. Effector cells carry out the elimination of the antigen. In the body, helper T lymphocytes activate other lymphocytes and phagocytes, uh, whereas regulatory T cells keep these cells in check, meaning they prevent any exaggerated immune responses to occur. Cytotoxic T lymphocytes, macrophages, and other leukocytes function as effector cells in different immune responses. So those effector cells, again, accomplish the final stages of the immune response with elimination of the antigen. So things, again, like activated T lymphocytes, phagocytes, and other leukocytes within the immune response. So regulatory cells are orchestrating and controlling this response, either turning on or off different functions in the immune response. And the effector cells carry out the actual destruction and elimination of those antigens. So we also within this immune system have something called major histocompatibility complex modules, or MHC. So the MHCs enable the lymphocytes to discriminate between molecules that are native to the body, meaning self, and those that are not and may be harmful. So again, this is part of that self versus non-self regulation that is essential in the immune response. The class 1 and 2 MHC genes are responsible also for encoding what we call human leukocyte antigens, or HLAs. These are proteins found on the cell surfaces, and they define the person's tissue type, so define the host's tissue type. Each person has unique MHC proteins. It is almost impossible for any two people to have the exact same MHC profile. And these HLAs, or human leukocyte antigens found on the cell surfaces, are the major target involved in organ transplant rejection. So when someone gets an organ transplant, one of the things that we have to do throughout the span of that transplant lifespan is to suppress the immune system enough to prevent the host from essentially attacking the transplanted organ because of that difference in HLA proteins on the surface of the cells. So the class 1 and 2 MHC genes also encode proteins that play an important role in antigen presentation. So what happens is protein fragments from inside the cells are displayed by the MHC complexes on the cell surface. And this allows the immune system to differentiate between the body's own tissues and foreign substances. So cells which present unfamiliar peptide fragments on the cell surface are then attacked and destroyed by the B and T lymphocytes. The MHC1 complexes are cell surface glycoproteins that contain a groove, which is able to accommodate and hold a peptide fragment. MHC1 molecules then interact with our C cytotoxic cells and activate them when presented with foreign antigen peptides. MHC1 complexes are also present degraded viral protein fragments from infected cells. Class 2 MHC proteins have a groove or cleft that binds fragments of antigens as well. However, they bind fragments from pathogens that have been engulfed and digested during the process of phagocytosis. So what we see here in the diagram is a picture of kind of what I just described. So here, kind of on the bottom right, we have a cell that has been infected by a virus. So that virus is inside the cell. And on the surface of that cell, we have these MH1 or MHC1 molecules. And as the cell kind of breaks down parts of that virus, it presents a small peptide, or essentially a small piece of that virus that has infected the cell on the surface of the cell attached to these MHC1 proteins. And our CD8, or our one of our T lymphocytes, this is a T lymphocyte, can recognize and bind to that MHC with the presented antigen peptide. And once it binds to it, it actually can go ahead and kill or destroy that cell. So this presentation of these antigen peptides on the cell by our MHC molecules is what signals this T lymphocyte to attach itself and destroy this infected cell. Similarly here on this phagocytic cell, so this is a cell that has phagocytized a invading antigen. So this is one of the cells of the immune system, and inside of it is now this phagocytized antigen. And so small peptides from that phagocytized antigen are then displayed on the surface of that cell by our MHC2 molecules. And that allows our CD4 helper cells to bind to that cell. And then the CD4 helper cells actually go ahead and activate things like B lymphocytes, which can release antibodies in response. So cells known as APCs, or antigen presentation cells, or antigen presenting cells. So APCs must be able to express both classes of MHC molecules. The antigen presenting cells include things like dendritic cells, monocytes, macrophages, and B lymphocytes. So they capture antigens and then enable their recognition by the T cells, either CD4 helper cells or CD8 cells. And the antigen presenting cells initiate adaptive immunity. So they are also a bit of a bridge, between that innate and adaptive immune system, which we talked about a little bit earlier with kind of the DCs. But they help, we have the phagocytic cells or things that present those antigens when they're encountered in that innate system, and then they go ahead and they initiate that adaptive immune response. So this is just another kind of pictorial sort of description of that pathway for immune cell participation in that immune response. So up at the top, you see an APC kind of right here, right? Probably some kind of phagocytic neutrophil, and it has engulfed and presented an antigen here on its surface of that MHC, right? So here we have the cell fully engulfed in antigen. These little particles then attach themselves to those surface MHC2 molecules, and that attachment allows our T helper cells to bind to that receptor and then become activated, okay? So that little kind of T helper cell then goes on to further activate either more T helper cells or our B cells. And then you'll also see here that there's that kind of presence of cytokines to enhance that process. Once the B and T lymphocytes are activated, they become amplified in their action by the cytokines that are also released as part of that innate response. And then you can see that the lymphocytes then divide themselves several times to form these different populations of clone cells. And those clone cells continue to differentiate into several more types of effector cells, as well as into memory cells. So in the adaptive immune response, the effector cells destroy the antigens, and then those memory cells are retained. And that allows us to have that ability to target an antigen really quickly during a future encounter. So you can see that here now that this B cell is activated by T helper cells and cytokines, it starts to clone itself. It starts to make the plasma cells that will produce antibodies to directly try to kill those antigens, but then it also produces some of these memory cells that'll stick around for the next encounter. And same thing that you can see here with the cell-mediated immunity and our cytotoxic T cells. We do make some T memory cells that stick around for the next encounter. And then we also make cytotoxic T cells that bind themselves to a cell that contains some sort of intracellular antigen and then kills the cell. Okay. So this is a diagram of kind of what we were just talking about as far as memory cells and their ability to allow us to um, react more quickly if an antigen is encountered again. So in that system of humoral immunity, the primary response of a body to an antigen is to trigger B cells to proliferate and differentiate into plasma cells that produce antibodies. And this process goes on for about 1 to 2 weeks. And the number of antibodies that we're making continues to rise even after the infection is completely resolved. And some remain intact to form this kind of pool of memory B lymphocytes. So you can see that here. So we have a naive B cell that has not encountered an antigen yet. But when we introduce that antigen, we have this primary response, and we get this big rise in the activation of B cells. Um, and then, as you know, we've cleared that infectious agent, so that primary response starts to ebb, but we still have these memory cells around. So if that antigen is encountered again, we have this much more rapid rise in the activation of B cells, so much more exaggerated and rapid response the second time we encounter an antigen. So secondary response occurs when that same antigen is encountered again, and antibody levels rise sooner and reach higher levels due to the availability of those memory cells. This is also the process that immunization makes use of in both the primary and secondary phases of humoral immune responses. So I mentioned earlier that we call the antibodies produced by the B lymphocytes Igs. So we have 5 different classes of Igs or immunoglobulins, and they have different functions. So the IgGs primarily function to display antiviral, antitoxin, and antibacterial properties. IgGs are responsible for protection of newborns. They activate the complement system within the immune response, and they can bind to macrophages. The IgAs are the predominant or most common Ig in body secretions. So they help to protect the different mucous membranes. So we find IgAs in the mucous membranes in different bodily secretions. IgMs form natural antibodies. They are the prominent immunoglobulin in early immune responses, and the IgMs will help to activate the complement system. IgDs are found on B lymphocytes, and they are needed for the maturation of B cells. And finally, IgEs. IgEs bind to mast cells and basophils. So IgEs are largely involved in things like parasitic infections, allergic reactions, and hypersensitivity reactions, which we'll talk about in our next unit. So a little bit more about the T lymphocytes. So functions of the T lymphocytes. They can activate other T cells and they can activate the B cells. They largely control intracellular viral reactions. T lymphocytes are also responsible for things like rejection of foreign tissue grafts, so during things like organ transplant. And they also play a role in something called a delayed hypersensitivity reaction. So different types of T cells. So we have helper T cells and cytokines. So activation of T cells is a central event in the initiation of both humoral and cell-mediated immune responses. So if you remember back to that diagram we looked at, when we had that presented, that APC cell, the macrophage cell with the engulfed antigen, it was the T helper cells that responded first to that APC, right? And that response by those T helper cells initiated the rest of that humoral and cell-mediated immune response. So those CD4 plus helper T cells are really this kind of master regulator of the immune system. They also secrete cytokines that help to influence function of nearly all the other cells of the immune response. That cytokine release determines whether a humoral or cell-mediated response will occur. So these helper T cells, these CD4 cells, are an absolutely essential component for a healthy and functioning immune cells because they really just sort of initiate the entire process. And we also have regulatory T cells or Tregs and cytotoxic T cells. So Tregs control immune system responses. They can suppress immune activity to try to limit the inflammation and tissue damage that's happening overall. So essentially, we need Tregs to shut this system off as well. We can't, again, have these systems just kind of running without any sort of control, or we start to get a lot of tissue damage. The Tregs sort of take care of that. And then we also have these cytotoxic T cells, or our CD8 cells, and they monitor cellular activity and destroy any cells that threaten the integrity of the body. So again, here's our little activated T helper cell, our little CD4 cell. It releases cytokines that activate that cytotoxic T cell. So you can see here's my cytokine receptors, that IL-2 interleukins, that's a type of cytokine. This cytotoxic T cell has receptor sites for those released cytokines. And when cytokines bind to those receptors, it activates the cytotoxic T cell to bind itself to that presented antigen on the target cell and essentially destroy it. The cytotoxic T cells can also release other cytokines that help to kind of perforate or destroy the cells as well. So again, those cytotoxic T cells recognize antigens on the cell surface that are presented by those MHC cells, those class 1 MHCs, to destroy that cell that's infected with either a virus. They can also bind to and attack cells that have been maybe transformed by different cancers. So the immune system plays a predominant role as well in the control of different types of cancer formations in the body. And they destroy those target cells through a variety of different mechanisms, but the end result is destruction of that target cell by those cytotoxic T cells. So again, cell-mediated immunity, that T cell-mediated immunity, requires healthy CD4 and CD8 T lymphocytes to be able to function. And that overall release of cytokines by the CD4 helper cells enhances the function of the cell-mediated immune response, the cytotoxic T cells, and those effector macrophages. And these cells also play a role in things like delayed hypersensitivity reactions, like what we call allergic contact dermatitis. And we'll, again, talk about that a little bit more in our next unit. So we mentioned earlier the lymphoid organs and their action in things like storing and movement of the different lymphocytes. So we have central lymphoid organs, which is the bone marrow and the thymus, and these provide the environment for immune cell production and maturation. Remember, the B cells mature in the bone marrow and the T cells mature in the thymus. And then we also have the peripheral lymphoid organs, and their function is really to trap and process antigens and promote the interaction of those antigens with those mature immune cells. So the peripheral lymphoid organs are things like the lymph nodes, spleen, tonsils, and appendix. And again, they serve as sites to enhance that interaction between antigens and the immune cells. So within the immune response as well, we have both active immunity and passive immunity. Active immunity response is a specific protection induced by that exposure to antigens. But in passive immunity, this is when specific protections are induced through transfer of protective antibodies against an antigen. So where we're transferring antibodies from one source to another. So a common example of this is the transfer of IgG antibodies across the placenta that protects newborns during the first few months of life. So that newborn has not produced any really antibodies of its own yet. So it really relies on transfer of antibodies from the mother for protection. And then also we transfer something, or we transfer IgA antibodies in the colostrum. So that's in early breast milk. So that's really how we get this passive immunity, especially in those early stages of life. As we develop an immune response, during fetal development, immunity starts to develop as early as 5 to 6 weeks. Um, the secondary lymphoid organs are really well developed after birth, and we start to produce IgA and IgM antibodies shortly after that time. And they will reach their adult levels by approximately 1 year of age. But if we're thinking about this, these things are well developed and we're starting to produce some antibodies. But throughout that kind of first year of life, we do consider an infant's immune system to be relatively immature and do consider them to be really at higher risk of developing infection for that reason. And as we age, of course, the elderly become more susceptible to infections. They also have more evidence of things like autoimmune and immune complex disorders. And they also have higher incidence of cancer than younger people. And all of those things are really tied to the overall kind of decline of the function of the immune system. So we have this overall declining ability to adapt to these environmental stresses. So there's a decline in our immune responsiveness. The size of the thymus gland really decreases as we age. And so if you remember, the thymus is where the T lymphocytes really started to mature. So we get less maturing T cells because of the loss of the thymus tissue. There's also a kind of an inborn biological clock in those T cells. So they start to kind of die off and their numbers are naturally reduced as we age. And there's also an altered response to the immune cells to antigen stimulation. So that kind of response to antigens isn't as, I guess, I don't know what word I'll use, maybe forceful or efficient as it is in younger people.